Welcome back. This video is for chapter 18, Blood, and this starts out a whole section where we will go through blood, the heart, and a little bit more detail about blood vessels and circulations, and I believe that's 3 chapters all on the same subject, blood and circulation. All right. So blood is a form of connective tissue, as we talked about way back in Chapter 5 at the beginning of last semester. And it functions to transport a number of things, including gases like oxygen for cell respiration, and it's bringing it to your cells. And then it's transporting away from your cells carbon dioxide, which is a waste product of cell respiration. And then it's also bringing nutrients, transporting hormones, and transporting wastes away from your cells. It also functions to help to regulate your temperature, and I think we talked a little bit about that in the integumentary system, so you have some control over vasodilation and constriction to the surface of your skin, and when you vasodilate and bring that blood to the surface of your skin, that allows your body heat to be released more into the environment. And so when you constrict it, it keeps it deeper in your tissue, and that conserves your temperature. Also has a role in pH balance, which we'll talk about more later, and fluid balance as well. And then we'll see that you have a number of things in your blood which offer protection against pathogens, and that includes your leukocytes or white blood cells, as well as a number of plasma proteins we'll be looking at in this section, as well as later on when we talk about the immune system. And blood also has a number of things that prevent blood loss, like we'll see platelets and then several plasma proteins as well. The adult has about 5 liters of blood, and that depends on your body size. So bigger people have more blood, smaller people have less, and in general men have a little bit more blood than women. And blood is thicker than water. It's about 4 times as viscous as water. And if you've ever lost a large amount of blood and you can feel it coming out, you can feel that that blood is warm. It's about 1 degree Celsius warmer than your body. And blood is slightly alkaline. It has a pH somewhere between 7.35 and 7.45, so that is above neutral, which is 7. We're going to see that arteries are always transporting blood away from the heart. Most often that's oxygenated blood, but not always. The thing that makes it an artery is that it's transporting blood away from the heart, and it's therefore going to be under pressure. Veins transport blood towards the heart, oftentimes is deoxygenated, but the pulmonary veins, for example, are oxygenated. The thing that makes it veins is that they're transporting blood towards the heart, and consequently they are not under pressure, and they have a structure that reflects that. Connecting to between your arteries and veins are capillaries, which allow that exchange of gases and nutrients, hormones, etc., between your blood and your body tissues. You can look at the components of blood by centrifuging a blood sample, and if you do this, it will separate blood into 3 main components, and it's separated by weight. So the top portion here is plasma, and plasma contains mostly water, some plasma proteins we'll talk about a little bit more later, like albumins, globulins, fibrinogen, and other dissolved proteins. And then a small amount of other solutes, like other ionic compounds or electrolytes, respiratory gases like carbon dioxide and CO2, nutrients, and waste products. So anyway, the plasma, the top part, is mostly water and proteins, plasma proteins. Then this little tiny region in the center here is called the Buffy coat. And this contains all of the formed elements other than red blood cells. So here we find platelets and leukocytes, which is just another way of saying white blood cells. And we'll talk about the different types of leukocytes later. And then finally, the most numerous solid portion of the blood gets stuck at the bottom after you centrifuge, and these are the erythrocytes. And erythrocyte is another way to say red blood cells. And these are the guys that transport oxygen as well as some carbon dioxide. So if you look at these terms here, whole blood represents all of this stuff together. It's your plasma, which is a liquid component, plus your formed elements. And the formed elements, again, are red blood cells, white blood cells, and platelets. And the reason they're not called cells, the whole group of them, is because red blood cells and platelets are modified kind of portions of cells. A related term you could look at clinically is a hematocrit, which is the percent volume of a patient's blood that is composed of red blood cells. So that's one way to look at blood. Another way to look at blood would be by making a blood smear. And this is something we would normally do in the lab. We're not going to do this semester just because we don't all have microscopes and you really can't see much without a microscope. But it's not a hard thing to do. You take a prick of your finger or wherever else and put that drop of blood onto a slide and take another slide and just spread it out all over the surface of the slide. And then you would stain it with a stain usually that will be called Wright's stain. That's just to allow you to see the elements a little bit better. But even if you didn't stain it, you would see your red blood cells at least very clearly. The stain helps more with seeing the white blood cells or the leukocytes like these guys here. And you can see that the most numerous thing, of course, are the red blood cells, or the erythrocytes, and there are far fewer white blood cells. And you can see also that the white blood cells are bigger, and they have these prominent nuclei. They have these weird, oftentimes multi-lobe nuclei, like this neutrophil here. And then you can also see here and there some platelets, and the platelets are very, very small. They are just little broken off pieces of a cell. And that cell is called a megakaryocyte, as we'll see later. So by making a blood smear, you could check a patient's number and the shape of their red blood cells, white blood cells, and platelets to see if they're normal and get clues to their clinical condition. So as I mentioned earlier, the blood plasma contains a number of proteins, and these proteins are called plasma proteins, right? Makes sense. They're proteins in the plasma. And these proteins are produced in the liver, except for your immunoglobulins, which are also called antibodies. And those antibodies are produced by B cells, which usually are housed in lymphatic structures, so we'll see later. So this list of your major plasma proteins here is by weight or percent volume. And so albumin is a major plasma protein. And one of the things that it does is it provides osmotic pressure in the plasma. And so when we talked about osmosis, the way to increase osmotic pressure would be to increase the number of dissolved particles or to decrease the amount of fluid. So anyways, by having dissolved components in your blood, that means that the blood as a result of having many dissolved particles, is going to draw fluid into itself, and so that's going to help to keep the fluid in the blood. Also, albumins function to transport various hormones and ions, as well as some lipids. Excuse me. Globulins, I mentioned you have antibodies. Those are not produced in the liver. They're produced by B cells, as well as some transport globulins, which are produced by the liver. Fibrinogens form little nets that function in blood clotting. And then there is a small amount of other plasma proteins, which we're not going to get into. The cellular component of your blood are called formed elements. And again, they're not called cells as a group because the red blood cells are modified cells. They do not have nuclei or organelles, and then the platelets are just little broken off pieces of a larger cell that are called megakaryocytes. But anyways, the formed elements include erythrocytes or red blood cells, platelets, and leukocytes or white blood cells. The process of producing any of these cells is hemopoiesis. So that's a production of any blood cell, not just red blood cells. And all of these are going to be formed in your red bone marrow, which, as we talked about during your development as a child, that's all throughout the majority of your bones. But as an adult, it's mostly inside of your axial skeleton, and then the head of your femur and the head of your humerus. So this process of hemopoiesis is taking place in red bone marrow and starting out with a type of stem cell that's called a hemocytoblast. And the hemocytoblast is going to be the stem cell that gives rise to all of your other formed elements. So if you look at this cell lineage here, it's pretty complicated. We start with this hemocytoblast, and then we go down one of two lines, the lymphoid line or the myeloid line. And what are we talking about here with lines? Remember that all of your cells are genetically identical. So these particular stem cells in your red bone marrow that are called hemocytoblasts already have a pattern of gene expression that will allow them to develop down either the myeloid line or the lymphoid line. And then depending on which line they go down, they're going to express different and different genes until you end up with all of your formed element types. Anyway, following down the myeloid stem cell line, you see that you end up with red blood cells, platelets, and then these different leukocytes here, basophils, eosinophils, neutrophils, and monocytes. Following down the lymphoid stem cell line, you end up with lymphocytes, which are B cells, T cells, and NK cells. So I've tried to simplify this diagram here to what I think is the essential information. So again, you start out with the hemocytoblast. This is a stem cell in your red bone marrow. And this starts to differentiate into one of two lines, either the myeloid line or the lymphoid line. The myeloid line further develops into erythrocytes, and that development is stimulated by EPO from the kidneys. That's a hormone we talked about previously. Or they become leukocytes, which are all of the white blood cells, with the exception of lymphocytes. And then they also become megakaryocytes, which are these big cells here, which are going to produce platelets. Down the lymphoid line, we get your lymphocytes, which are your B cells, your T cells, and your NK or natural killer cells. Probably the easiest way to remember this is that your lymphoid line, as the name suggests, lymph produces your lymphocytes. And then the myeloid line produces everything else. So let's take a look at these cells individually in a little bit more detail, starting with your red blood cells or your erythrocytes. They function to transport primarily oxygen, and they also transport some carbon dioxide. As I said before, these are modified cells. They do not have a nucleus, and they do not have organelles, and as a result, as they break down over their 120-day or so lifespan, they cannot self-repair. They can't fix themselves. So they get destroyed in the liver and the spleen. And they have this characteristic donut type of structure, which is called biconcave, because one side is concave and the other side is concave. And they're also sort of flexible so that they can stream through your capillaries. And they, at the narrowest parts of your capillaries, they go through one by one. So it's very thin in there. And so they have to have this particular shape and this flexibility to allow them to move through. In sickle cell anemia, for example, they do not have the same flexibility, and that can cause them to get stuck in the blood capillaries, and this can lead to stroke and other related problems. Within your red blood cells, you have this molecule called hemoglobin. And a hemoglobin is a protein that is made of 4 globular groups. So you can see here, here's 1 globin, 2, 3, 4. So there are 4 globins, and the center of each of these globins has this heme group, and that heme group they've zoomed in on here. And the really important thing about the heme group is that in the center of the heme group you have iron ion. And again, you have 1 of these per each globin, so you're 4 heme groups per hemoglobin. And the reason that that iron group is important is because oxygen binds to that iron, and that's how oxygen is transported in your blood. So you breathe in oxygen, and that comes close to your red blood cells at the capillaries of the alveoli. Oxygen gets picked up by the hemoglobin within your red blood cell, and then the red blood cell is transported through circulation to your tissue cells, where oxygen will then be released. CO2 is also capable of binding to hemoglobin, and instead of binding to the heme group, it binds to the globin protein, but only about 23% of CO2 is transported this way in the blood. Most of the CO2 has this reaction with hydrogen atoms, and it ends up being dissolved and transported as this bicarbonate. I have a sneeze, or carbonate molecule. So anyway, the majority is transported, dissolved. As I said before, EPO from your kidneys stimulates the production of your red blood cells. And the stimulus for EPO production is a decrease in blood oxygen levels, or it can also be stimulated by an increase in testosterone. So as a result of that, men usually have a higher red blood cell count than women. So what would reduce your blood oxygen levels? Well, if you're losing blood, for one, or if you're at a high altitude. So, sometimes elite athletes will train at a high altitude in order to decrease their blood oxygenation. That causes the release of EPO, which subsequently causes an increase in red blood cell production, so that when they come back down to sea level, they have all of these extra red blood cells that can transport more oxygen to their muscle tissues, so they can perform, their sport with a greater level of endurance. And also, if you have your red blood cells removed, like if you're donating blood, that will also stimulate EPO release. So that's going to be released by your kidneys. Your kidneys release EPO into the blood. It is a hormone, so it travels in the blood. And that's going to be picked up by cells in your red bone marrow. And it's going to stimulate the myeloid cells to make more red blood cells. And this is something that is illegal to do, but many athletes take advantage of this. And this is called blood doping. And there's a couple of different ways to do this. One is basically by donating blood to yourself. And so what happens, you remove your blood prior to your competition, and then you store that blood. And after some days or weeks, you're going to have an increase in EPO levels, and then a production of red blood cells, so your red blood cell level will be back to normal. And then you will inject your red blood cells that you previously stored back into yourself, so that then you have your normal red blood cells in your body plus the red blood cells you have donated back to yourself. And then the other thing that people do is they take pharmaceutical EPO, synthetic EPO hormone, and that's going to cause our own bodies to produce more red blood cells. The red blood cells were the primary things which gives blood viscosity. So by blood doping, you increase your blood viscosity, which means your heart is going to have to work harder to pump that blood through your body, and that can cause permanent cardiovascular damage. Okay, so like I said before, red blood cells are being recycled or destroyed in your liver and spleen. And that is because the red blood cells cannot self-repair. And so they have a limited lifespan of about 120 days. So they get produced, in your red bone marrow, as we have previously discussed. Then they go through your circulation for about 120 days. And then finally you have macrophages in your liver and spleen that are going to break down those red blood cells. The iron from the red blood cells is going to be stored in the liver and spleen, and then it'll be transported back to your red bone marrow when you need it to make more red blood cells. The heme group is used to make bile, and then the amino acids are going to be recycled to build new proteins. Okay, so moving on to blood types, and we'll test our own blood type in the lab for this section. And blood type really just depends on 3 antigens, A, B, and Rh, which we'll look at in just a moment. We'll start out here with the ABO blood type, and the ABO blood type depends just on the A and the B surface antigen. And an antigen is just any molecule which can be recognized by an antibody, and usually, but not always, usually they sit on the surface of a cell, and in this case they do sit on the surface. So either you have cells like this with A antigens, cells like this with B antigens, or you have a cell with both A and B antigens. So this guy here with a surface antigen A is called type A. So the type refers to the antigen, the cell surface marker on your red blood cells. You don't produce antibodies to your own antigens, or they would attack your own cells. So a person with type A blood has type B antibodies. And so those antibodies will not attack their own blood. So type A has A antigens and B antibodies. Type B has B antigens. And then again, because you don't want antibodies that attack your own cells, they produce A antibodies. Okay, and then the type AB blood has type A and type B antigen, and they do not produce either A or B antibody because either of those antibodies would attack their own red blood cells. And then finally, type O blood does not have A or B antigens, and as a result, they're going to produce both A and B antibodies. So if you forget, what all the blood types mean, all you really have to know is that the type refers to the antigen on the surface of the red blood cell. So again, let me just say, for example, if you're looking at type B, that means this has B antigen. And then it has the opposite antibody because you don't want antibodies that are going to attack your own cells. Okay, independent of the AB antigen, you also inherit this Rh factor. And so you've probably seen, just to continue with this example here, this could be type B, and then it would be positive or negative. And that positive or negative designation refers to the Rh factor, which unfortunately is also known as the D antigen. It's the same thing. So this is really not any more complicated. If you're Rh positive or Rh plus, that means that the Rh or D antigen is present. I'm just going to go D because it's easier to write. So that would be Rh positive. If you're Rh negative, that means you do not have the Rh antigen. The thing that is different about this is that the antibodies to that Rh antigen only appear in the blood if you are Rh negative and you've been exposed to somebody with Rh positive blood. So that those antibodies are not normally floating around. And that becomes important, particularly for women, because if you're Rh negative and you're carrying a baby that is Rh positive, like this guy here is Rh positive, well, normally your blood and the baby's blood are separated from each other, but as it says here, the baby's blood could come in contact with your blood during delivery. And so if you're Rh negative and you get that exposure to that Rh positive blood, you're going to make this anti-Rh antibody. And that will be a problem if you have another pregnancy and the next baby is Rh positive, because those antibodies, the Rh antibodies, can cross the placenta and they can attach to and damage the baby's red blood cells. And the way that you can test for blood type compatibility is through a simple test called red blood cell or erythrocyte agglutination. And what will happen is that red blood cells make clumps when they are cross-linked by antibodies. And by the way, it's not just red blood cells, bacteria, viruses, many things that antibodies would bind to, the antibodies will cause them to clump together. So that's a problem clinically if you have clumps of red blood cells like in this guy has here from his blood transfusion. That's going to clog up his small arteries and veins, and particularly the capillaries, and that could lead to stroke or similar problem. So that agglutination depends on the donor's antigens and the recipient's antibodies. And I hope this is going to make sense why. So before you go transfusing blood into somebody, you could put the two types of blood together on a slide or in a little petri dish and check to see if the blood will agglutinate. Okay, so let's see here. This is a type B recipient. And I said it depends on the recipient's antibodies. So here is this type B because he has B antigen. This is the person that's going to receive the blood transfusion. And this person has A antibodies. If we take blood from a type A donor, this person has A antigens. By the way, he also has B antibodies. But that doesn't really matter because when we take the blood, we're just going to take, in the transfusion, we're just going to use the red blood cells, so the antibody of the donor doesn't matter. So when we combine these things together, we're going to get this person that has B, their original, what am I doing? They have their original type B blood, and those guys are going to be fine. Those red blood cells would be fine. They do not interact with their own A antibodies. But since you've added this type A blood, the A antibody that was already present in your recipient is going to bind to the red blood cells and cause them to clump together. So like I said, you can do this in a dish to test for problems ahead of time. So like here is I don't know if it's the same example, anyway, similar example. The donor has type A blood, and you're adding that to a recipient that has type B blood, so this person has the antibody for that A antigen, and that antibody is going to cross-link the red blood cells and cause them to clump together, and you could see that clumping just with the naked eye. Whereas if you have a successful match of blood types, there's not going to be any clumping, and it'll look more like this image here. So this not even through a microscope. You can just put it in a small well dish, and you'll see that reaction pretty clearly just with the naked eye. Okay, moving on to leukocytes, or white blood cells. Your white blood cells function in immune defense. And there's a couple of different ways to look at these, and I'll try to do a few. You can divide them into granulocytes and agranulocytes. Agranulocytes. And this is just like a visual distinction where after you do the right stain, if you look at them under the microscope, these guys at the top here look like they have granules. Well, they do have granules. They're visible granules. And you can see these little dots inside of them. And that's what makes them granulocytes. The agranulocytes don't have visible granules, or the granules are less visible, and so they are agranulocytes. I really don't like the names because they don't really have much to do with the function or the lineage of the cells, but they are still commonly used, and you could use this, if you're doing a blood smear to help you to identify these different cell types. Anyways, the granulocytes include neutrophils, eosinophils, and basophils. Neutrophils are the most common type of white blood cell. These are the kind of first responders. They're generic phagocytic cells, so they're going to phagocytize any pathogen, any bacteria, any viruses that come in that do not belong in your body. They are not specific. They are a form of non-specific defense against any foreign agent. And they also they have a reaction which tends to kill them in the process of killing the pathogen. And the dead neutrophils form a large portion of that fluid that you see that comes out, in pus if you have an infection. If you did a blood smear and you counted the different blood cells that were in a person, you would see that neutrophils are likely to be increased if that person has a bacterial infection. Eosinophils act in parasitic worm infections, and their numbers will also be up during allergic reactions. Basophils act in inflammation, and they release histamine, which causes vasodilation and is responsible for a number of the symptoms of allergies. Lymphocytes, remember, are your B cells, T cells, and natural killer cells. They function in cell-mediated immunity, which another way of saying that it would be antibody-mediated immunity or specific immunity. And these guys are going to usually be increased if you have a viral infection or if you have a chronic bacterial infection. Finally, you have the monocytes, and I've written down here monocytes, macrophages, dendritic cells, because a monocyte can differentiate into either macrophages or dendritic cells, and these are all three types of professional phagocytes, or they're also called professional antigen-presenting cells. And these guys, these antigen-presenting phagocytes, they will eat up the pathogen, digest it internally, and then display a piece of that antigen, of that pathogen rather, on their cell surface. So they present that to other cells, to T cells and B cells, to activate the adaptive immune response. So they are professional APCs, antigen-presenting cells, because they present antigens to B cells and T cells to activate your specific immune response. as I said before, you could look at differences in the types of cells that are present by doing a blood smear and get these differential counts. So leukopenia would be a low number of leukocytes, and if somebody has low number of leukocytes, they're at an increased risk of infection. Leukocytosis would be an increase in leukocyte count, and that could be caused or will be evidence of a recent infection or stress. Neutrophilia, as I mentioned before, an increase in neutrophils is associated with bacterial infections. Could also be stress or tissue necrosis. Neutropenia, or a decrease in neutrophils, is associated with anemia or various invasive therapies like radiation therapy. Leukemia is a type of blood cancer that usually begins in the red bone marrow and results in a increase in the number of abnormal leukocytes. So you make all these leukocytes that do not carry out their normal function. As a result of that, you're going to have a decrease in your erythrocytes and megakaryocytes, which make platelets, and you're going to have also a decrease in the number of normal functioning leukocytes. And as a result, you can develop anemia, bleeding, and an increased risk of infections. Often in children, this is acute, and it can progress rapidly and can lead to death within months. If it's happening in older people, and you get that chronic leukemia, that progression can be much slower. Okay, so we went through red blood cells, white blood cells. Now the last one we'll come to are the megakaryocytes, which produce platelets. So like all of your other all of your other blood cells, they're produced in red bone marrow. The megakaryocytes themselves never leave. Instead, they shoot out these little extensions through the epithelial cells of your blood vessels that are called protoplatelets. And as the blood rushes past them, it rips off little pieces of those protoplatelets to make platelets. So platelets are cell fragments of megakaryocytes, and they're going to be important in hemostasis, which is stopping bleeding. And that process of stoppage of bleeding, or again hemostasis, occurs in 3 phases, which you can remember as squeeze, plug, clot. So the squeeze part, the first part, is a vascular spasm. You have smooth muscle contraction, which limits blood flow at the area of injury. So here this person is getting injured. The blood vessel here is going to squeeze, or the smooth muscle in the blood vessel will constrict to close it off as much as possible. And that's going to last anywhere from on the range of minutes. So fairly short lasting. The platelet plug also does not last very long, also minutes. And here the platelets that are normally rushing by in your normal circulation are going to stick to the exposed collagen fibers, which have been exposed by the injury, and they attract other platelets to stick as well, and so they form a platelet plug. Helping them to stick together is a protein called von Willebrand factor. And then finally, a longer lasting blood clot will occur through the process of coagulation. So there are a number of enzyme cascades which occur in that platelet plug, which are going to convert inactive forms of proteins which are normally present into active forms, which in the end activates a protein called fibrin, and that fibrin is what you see here making this net, and that fibrin traps other formed elements in that net, like platelets, red blood cells, etc., to form that clot. Well, here's a cool electron micrograph picture of that fibrin clot. So here's the fibrin protein. There's a kind of spiderweb strands, and they're trapping these platelets as well as the erythrocytes to form that clot. The last thing we'll talk about is the sympathetic response to blood loss. So if your clot is not successful in stopping your blood loss, or for some reason, any other reason you're losing blood, if you have lost more than 10% of your blood, the sympathetic nervous system will kick in and cause vasoconstriction, an increase in your heart rate, an increase in the force of contraction of your heart, and then a redistribution of blood flow to your heart and to your brains. So it may be counterintuitive that you're losing blood, that your heart is going to pump harder, but you need to make use of the small amount of blood that you have left to keep your tissue oxygenated. So you want to keep your heart and your brain alive while you're losing blood. And this sympathetic activation is going to be successful at maintaining your blood pressure until about 40% of your blood is lost. And then that's probably the end of the story for you. Okay, that's it for this whole chapter. I'll see you next one, and we'll move on to the next chapter.